topic by topic. We're going to have a look today at anaerobic respiration. We've looked at aerobic, and aerobic is where we can work with oxygen. Would you like me to start again? And what we're going to try and do is we're going to look at anaerobic today without oxygen. And I've got an experiment I need to set up now so we can see the results later on. What I've got here is a very simple setup. I've got a boiling tube, and into this boiling tube I'm going to put some lime water, and lime water is good for changing colour. It goes cloudy when carbon dioxide goes in. So what we're going to do is we're going to take, that went wrong, a measure of yeast, and I've got here some pelleted yeast, and what I'm going to do with this is I'm going to add to this some sugar. This is going to be messy, I can see that. Just plonk that over so all the sugar goes in. There we are, we've got some sugar and some yeast. I'm going to add a little bit of warm water to that, and then what I'm going to do with that, once I've done that, is I'm going to cover that with some oil. So let's just make sure we've got some warm water. I failed to make sure I had warm water before I started. And it's quite warm today, quite pleasant. So it's about 30 centigrade here. So I'm going to give this a really good mix, because what I want to try and do is get the yeast activated. And what we're then going to do is, because I want this to be anaerobic respiration, what I'm going to do is I'm going to put some oil over the top. So I'm just going to take some ordinary cooking oil. And I'm going to put a few squirts of this. Over the top. Now, doing this is going to try and make sure that any air in here cannot get to the yeast. I think we're just going to need to put a little bit more in. I think that should more or less do it. I can now see that I've got a layer of oil over the top of my yeast and sugar solution. So I had a little problem with the camera there. Right, so we've got our yeast and sugar, we've got oil over the top, and what I'm going to do is I'm going to put this into the lime water, put the bung on, and we're going to leave this for as long as we can to have a look to see whether we get any carbon dioxide produced. And if we've got carbon dioxide produced, it's going to turn the lime water cloudy. So I hope this is going to work. We'll see. Let's get down to business and have a look at what we've set up and what we're trying to do. Now, in this experiment that I've set up, we're basically looking at this type of setup. I've got here my yeast with sugar and water, and over the top of that we've put a thin layer of oil. Put a bung in the top, and then I've got a piece of glass tubing that's going to go into a boiling tube with some lime water in it, and hopefully that should bubble and we will get some carbon dioxide produced, and as we get some carbon dioxide produced, we're going to get this turning cloudy. Because let's look at what reaction we've got. We've got here some glucose, no air, so this is going to produce alcohol, particularly ethanol, plus carbon dioxide, and this is the reaction which we will call anaerobic respiration. And this is our, if you like, classic experiment to try and set up to see what happens. We can in fact change the sugar. I used in here sucrose, but we could have just as easily have used things like glucose or perhaps even better maltose. So we could use different sugars in here and we could see how that affects it. We can also change the concentration of sugar, and we can look at how temperature affects this reaction. So we can do a whole load of reactions and experiments with this basic setup, which I've just got set up there, hopefully going so we can sort of see this working a little bit later. But and as with most biological systems, this takes quite a long time. We can test for various different things here. I'm using lime water, but I could also use bicarbonate indicator. Using bicarbonate indicator, I can actually measure the colour change more accurately than I can with the lime water, and we can work out various rates. So this is our basic experiment, and we'll see if we can deal with all of this. So there we are, we're ready to try and have a go and see what we've got. Now, once the experiment's finished, and we can't easily do this for you, I can actually remove the oil and we can smell it to see if any alcohol has been produced. Of course, smell doesn't work terribly well on YouTube. Now, what we've got also here is some other basics of experiments. So this is great for making wine and beer, but it's also excellent for making things like bread. And what we do is we make that nearly every day. Make the bread by putting in yeast and sugar and it fermenting quite nicely. So I'm hoping that this should work. We'll see. So anaerobic respiration. We can look at anaerobic respiration in animals and in plants. Now, in plants, what we expect to see is our glucose turned into alcohol, which is ethanol plus carbon dioxide. In animals, that isn't true, and in animals we make lactic acid, sometimes just called lactate. This builds up and this then needs to be got rid of. So let's have a look at this sort of experimental sort of system. So let's start by having a look at lactic acid. Now, there are various experiments we can do with this, and these aren't particularly easy to sort of have a look at and do, but they are ones which you may well be familiar with. So what you might do is you might have to do some strenuous exercise, and when you do some strenuous exercise, we're looking at lactic acid building up in the muscles, and we find that it sort of hurts. That's one way of trying to do it. There's an easier way of trying to do this, and this involves taking a weight. Something like my mouse here is not suitable, and we hold this out at arm's length, and you hold something like a kilogram, 2 kilogram weight out at arm's length, and you'll find that the fatigue cuts in really quite quickly. You don't have to go clenching muscles and doing all sorts of things. It rarely seems to work, but holding out a weight in your arm, holding it out and trying to hold that for a minute or so becomes really quite painful as we start to build up the lactic acid. And you'll find that not just holding it out fully, but if you bend your arm very slightly, then this makes a difference. So the experiment that we would try to do is to hold out a large weight, and if I say that's something like a 2 kilogram weight, trying to hold that out, then after 1 to 2 minutes we start experiencing something called muscle fatigue. We see this in some runners sometimes, and there are some really quite interesting YouTube videos of someone nearly making it to the line and they can't quite, because what's happening is in the muscles, what we're doing is we're producing this lactic acid because there isn't enough oxygen. The lactic acid works on the muscles and it basically stops the muscles working. This is a good idea really, because as this moves up, we're running out of oxygen, we're running out of glucose, we're telling the muscles we're starting to run out of these things, we've got an enormous build-up of lactic acid. This needs to be got rid of, and so it tries to stop the muscles working, and this we call muscle fatigue. If we look at different sorts of muscles, we see this to be happening, and one that you may be familiar with is if we look at something like a chicken. Now, a chicken, most people are familiar with, has got basically two sorts of meat. We've got what's called the brown meat, and we've got what's called the white meat, and a lot of people prefer, especially kids, the white meat. The brown meat is slightly different. This contains a huge amount of blood vessels, and this mainly does aerobic respiration, and it's used in the things like the legs, which are used all the time, the back, and they're used to sort of, they're not doing an awful lot of exertion, but they're available and needed all the time. The white meat, which is used generally in the breast for flight, not used very much. So this has normally got a low or a lower amount of blood vessels, and when they're used, they're not used for very long, and typically these use anaerobic respiration. And as they use anaerobic respiration, then there is a build-up of this lactic acid which needs to be got rid of. And the word that we look here is something called oxygen debt. So we have a look at that. Now I'm just going to go back over to our experiment, and what we can see in this experiment is we're starting to get some bubbles coming through. So already I'm starting to get some coming through, and we know the yeast, which is down the bottom, can't be using any oxygen, and we're aware of that because we've got a layer of oil stopping it getting in. So it's got to be doing anaerobic respiration, and sure enough it's producing a gas, and we can see these bubbles coming through. So we'll leave this a little bit longer and we'll see whether this is starting to produce carbon dioxide and whether this lime water is going to go cloudy or not. So we've got this idea of oxygen debt. But what is it? Well, we go exercising. As we go exercising, our glucose is being metabolized, and this starts off being turned into carbon dioxide plus water. But rapidly we run out of the amount of oxygen that can get there, and so running out of oxygen, because you just can't get enough from the lungs, we start producing lactic acid. And this lactic acid basically starts to build up, and we start getting more and more lactic acid building up in the system, and this really isn't terribly helpful. So what we need to do is to remove this lactic acid, and it actually gets sent to the liver, and as it gets sent to the liver, there it gets converted back to glucose, and that glucose is then respired aerobically and produces carbon dioxide and water. But this whole process here takes time, and this is a big problem. So we need to, when we go running, have a look at a problem. So let's have a look at going running. And when we go running, what happens is we're not doing anything. We then looking at our basically oxygen, we sort of start breathing faster and faster and faster, and then let's say we stop doing exercise. So let's stop here. That's where we started, and this will be maintained, and then gradually will come down back to where we started. This here is all about paying back our oxygen debt. We were needing to use oxygen, but we needed perhaps that much oxygen, but we only had this much. We couldn't get enough to our muscles, so the muscles started producing lactic acid, and then as it's taken to the liver, then this is then respired later and turned into carbon dioxide and water. And so we have this paying back this oxygen debt. If we want to summarize this reaction, this is basically lactic acid plus oxygen give us CO2 plus water. Typically, if you go and run perhaps the 100-meter sprint, then basically many athletes don't even really breathe well at all, really. They don't really make an awful lot. So they make do with what oxygen is in the muscles already. And if you're only doing a 100-meter sprint, then there is plenty of oxygen here in the muscles to keep us going. But as soon as we get probably a little bit further than that, maybe a 200-meter sprint, then there may or may not be enough oxygen in the muscles. By the time we get on to a 400-meter sprint, then we certainly don't have enough oxygen in the muscles. And here we're having our lactic acid starting to build up because we're starting to do anaerobic respiration. But this typically is still not going to hurt us, because the amount of lactic acid built up here isn't going to make any difference really. But once we start doing something like our 1500 meters, or you want to do a 5k or a marathon, then we normally end up with here high amounts of lactic acid. This is why people train. They train so they can actually get fitter, so their body adapts better to this lactic acid build-up, so that the body can actually remove this lactic acid faster and send it to the lungs, get more oxygen to the lungs and send the lactic acid to the liver where it can be respired. So this is our paying back our oxygen debt. All of this comes really to do with something we'll call metabolism. What do we actually use our glucose for? We bring in our glucose, and in fact our glucose is normally turned into an energy transfer chemical called ATP, adenosine triphosphate. This is our currency for our energy, but our source here is glucose. Now, in our metabolic reactions, what we need to do is often to make glucose. So we can get our glucose, and that can come from a variety of sources, such as starch, such as glycogen, such as cellulose. All of those can be used to make glucose. But this reaction can go the other way. We could take our glucose that we've eaten and we could turn it into starch or glycogen or cellulose. We can take our glucose and we can form from this lipids, fat molecules, which are made from a molecule of glycerol plus fatty acids. We can take our glucose and we can add some nitrate and we can make the amino acids. Now, I'm talking here largely about plants. People tend not to make starch or cellulose. People don't tend to take glucose and turn them into fats. People don't tend to make their own amino acids. We've got also going down here, we've got our reactions of respiration. I suppose with this we can also put down, we've got our reactions of photosynthesis. We've got our breakdown of lipids into fatty acids and glycerol. And we've got our breakdown of proteins into amino acids. Push it up a little bit. All of these, whether they're in plants or whether they're in animals, these are the metabolic reactions. The reactions basically of life. And these reactions are required to make energy to move, to grow, to reproduce. this is what this energy is used for, all these sort of things and more. Let's just go off and have a look at our experiment again. Well, we've certainly got a lot of carbon dioxide being produced, and we can see here our lime water has indeed turned cloudy. So my anaerobic respiration experiment has actually worked, that we've got lots of carbon dioxide being produced, and this carbon dioxide we can see is producing a change here in the lime water, showing that we've got carbon dioxide. So I'm quite pleased with that one. That one actually worked. It's always nice when my experiments work. Let's see what's the role of the liver in all these metabolic reactions. The role of the liver is basically several-fold, but I suppose we think of it is I suppose detoxifying all the poisonous substances that you get in your body, especially those like ethanol. The liver is involved in the breakdown of old blood cells, so they are basically recycled, and they basically also break down products into the blood, so they can be sort of made into substances like urine, and that can then be removed via the kidneys. But the major function is dealing with the lactic acid. Let's, if we have a quick look at a liver, and let's just draw out a liver. The liver contains, oddly, two arteries going in, and we have one vein coming out. So one of these is called the hepatic artery. Now this hepatic artery brings oxygen from basically the heart and lungs. It brings in oxygen straight from the heart via the lungs. We've got a different artery that comes in, and this is called the hepatic portal vein. And at the moment you're saying, hang on a minute, that's a vein going into the liver. That doesn't sound right. Well, the hepatic portal, it comes from the intestines. So blood comes to the intestines, it picks up all the food, so the hepatic portal vein is bringing food, primarily stuff like glucose, and it's bringing it to the liver. And then blood leaves via the hepatic vein, and it returns to the heart with basically all the levels of food adjusted. So we take in lots of oxygen, lots of food, the liver processes this and then tries to send out a better leveled amount of blood round the rest of the system. So there is our basic role of the liver. The liver usually something like about, in a person, it's about 1.5 kilograms in weight, and it's really quite big. It sort of occupies really your sort of area around your sort of your body. Let's be more specific. If I've got here somebody's body, we've got the lungs coming in at the top, and the liver occupies this basic area with the stomach and a few other things, but it's a large organ covering the size of your sort of girth, I suppose. So we've got then the liver. Our next thing to finish off with is to look at what we can do with plants, and I'll spend a few moments having a look at what we've done with plants. So we've looked at simply here plants doing anaerobic respiration, and that is our glucose being turned into ethanol plus carbon dioxide. And we see this particularly in things like yeast. Yeast is good for bread, it's good for beer and wines, but yeasts aren't the only things that do this. There are in fact quite a few lactic acid bacteria, and you see adverts on TV for telling people to try and take some of this lactobacillus. This is a type of bacteria, and this will basically take milk and turn it into yogurt. And making yogurt is really very easy. First of all, we kill off everything in the milk, so we'll call that being pasteurize the milk. This involves heating it to somewhere anywhere between 85 and 95 degrees Celsius for somewhere between 15 and 30 minutes. Once we've done that, then we also, that's basically all it's done. It's killed all the bacteria in the milk. And then we cool the milk about 40 Celsius, and then we add our lactobacillus. So now we know we've only got lactobacillus in there, and that now starts producing lactic acid. It's starting to digest the milk, and as it does so, the pH becomes more acidic. It gets down to about pH 4, which is the optimum conditions for these bacteria to grow, and the mixture basically coagulates or thickens, and once it's done, then we cool to probably about 4 degrees Celsius, and we can keep it there, and that basically preserves everything. So we've got our yogurt ready to eat. Just in the same way that we've got bread, beer and wine, then we can make all sorts of clever sort of things to sort of go on, and this is the process of fermentation. So fermentation for things like wine, things like beer, and basically if you've ever tried watching someone or even had a go at trying to make this, it's quite interesting trying to sort of set this up, making homemade wine, making homemade beer, that we simply add a yeast to sterilized, well, I suppose even we consider beer, it's a sterilized fruit juice. We sterilize first to ensure that the bacteria that we don't want are killed off, destroyed, and then we add our particular yeast that we want, and this then does the reaction that we know, that we expect, and we see all the sugar turn to alcohol. The yeast that's left over, incidentally, for beer making, that can be hydrolyzed, basically killed off, and that makes stuff that you might be familiar with, that black goo Marmite, which you either love or, I suppose, as they say, you hate. Anaerobic respiration then. We've looked at a little experiment, the classic experiment to do that, and that can be modified in a whole variety of different ways. And we've had a look at the difference between animals, because they use lactic acid, and that's metabolized in the liver. I hope you've enjoyed this. I hope you found it useful. If you have, great, please subscribe, give us the thumbs up, and I'll see you next week when we'll do some more GCSE biology, topic by topic. Until then, stay safe. Bye bye.